The following is a selected video from masterthecontent.com, where you will find an extensive video library of lectures for a variety of standardized admission tests. We offer over 600 hours of detailed video lectures for a multitude of standardized tests. Use our interactive in-lecture table of contents to find specific topics of interest. Work through numerous in-lecture examples to help you internalize concepts. To learn more, visit masterthecontent.com. Your career, our passion.
Just going to move on, and we're going to look at a summary of this entire process. So it's very simple, and I'm just going to get you to focus or look at the enzymes responsible for these conversions and the substrates as well, which we are dealing with. Now, in the first step right here, we have our oxaloacetate actually being converted or combining with our acetyl coenzyme A to form a citrate, and the enzyme responsible for that process is citrate synthase. In the second step of our reaction, in the second step of this process, we have our citrate right here, which is step 2, being actually converted into our isocitrate. Now, this reaction involves an intermediary, which is referred to as cis isonotate, which is formed during that conversion, but the second step as a whole converts our citrate to our isocitrate. Now, in the third step of the reaction, our isocitrate is converted to the alpha ketoglutarate. Now, the enzyme that mediates that entire process is referred to isocitrate dehydrogenase, and the additional products that are formed as a consequence of this step will be NADH, obviously, and also a molecule of carbon dioxide. Now, the alpha ketoglutarate that's formed in this step is then converted to succinyl coenzyme A in step 4, and the enzyme responsible for that conversion, or for mediating that conversion, is the alpha ketoglutarate dehydrogenase enzyme. So that enzyme will form a complex that converts the alpha ketoglutarate to succinyl coenzyme A, and as well, we're also going to produce a molecule of NADH and a molecule of carbon dioxide. Now, in the fifth step, our succinyl coenzyme A, formed from the previous step right here, is then converted into a succinate, and the enzyme responsible for that is succinyl coenzyme A synthetase. It mediates the conversion of succinyl coenzyme A to the succinate, and the byproducts or the products of this step right here is a molecule of coenzyme A as well as a molecule of GTP. So you will remember that a phosphate group will replace the coenzyme A in succinyl coenzyme A, and then that phosphate group in that intermediary will then be transferred to GDP to create GTP, effectively forming our succinate in step 5. Now, in step 6, our succinate is then converted into the fumarate, and the enzyme responsible for mediating that process is succinic dehydrogenase, and that actually converts our succinate to our fumarate. Now, one of the products of this step, step 6, will be the FAD, or the hydroquinone that we were talking about. So it is actually going to dehydrogenate our succinate, and itself it's going to get reduced, and that results in the formation. It's going to oxidize our succinate, and itself gets reduced, which forms our succinate from our fumarate, and it also forms our FADH2 or a QH2, which is the same thing. Now, in step 7 of this process or this reaction, our fumarate is converted to our malate by the addition of a water molecule, which essentially rearranges the bonds in the fumarate to form that new or the next substrate in this process, which is the malate. Now, the enzyme that is responsible for catalyzing or for mediating this process is the enzyme called fumarase. So this enzyme right here, fumarase. Now, in the last and final step of the citric acid cycle, or the Krebs cycle, our malate is actually converted to, so this is step 8 right here, our malate is actually converted to the oxaloacetate, which is the first substrate in the reaction by the enzyme malate dehydrogenase. And that process actually, of that oxidation reaction, reduces a molecule of NAD+ to NADH, and the reaction is then free to proceed into another turn because the initial reactant or the initial substrate that we require, which is the oxaloacetate, has been reproduced, or has been regenerated as a consequence of all those individual reactions. So that's basically a summary of the citric acid cycle. I hope that's completely clear to you.